Now. Welcome to CoBAD, the Control of Breathing and Airway Defense International Online Seminar, sponsored by the Breathing Research and Therapeutics Center. We have two great speakers lined up today, but before we get started, just a couple of brief announcements. Today's talks will be recorded and they will be put online in perpetuity for now. So they should be there for some time if you want to go back and review things. Just as a reminder, the last session of season 4 of the CoBAD seminar series is August 4th. That'll be Brian DeLuey from the University of Iowa, and he'll be talking about the role of the unique portions of the brain and their role in suppressing breathing, particularly in things like epilepsy. As for now, our moderator today is Alicia Vose. Alicia is a newly minted assistant professor in the Department of Neurology at the University of Florida Health Science Center in Jacksonville. And after a number of years working as an SLP, particularly in Baltimore, Alicia came and finished her PhD here at the University of Florida in the Rehabilitation Sciences graduate program. We were then lucky enough to lure her to a joint project where Emily Fox was her main mentor as a postdoc, and I had the pleasure of co-mentoring her at that time. And Alicia is setting up a really interesting program looking at the effects of intermittent hypoxia on swallowing function, particularly in individuals with neurological injury, spinal cord injury, and TBI. So, Alicia, the show is yours. Thank you so much for moderating.
Great. Thanks so much for that, Gordon. I'm so excited today for obvious reasons, having two really great speakers in the swallowing and mastication world. I'm going to introduce both speakers today, starting with Dr. Lever, who is an associate professor in the otolaryngology department in the School of Medicine at the University of Missouri. As a clinician scientist, she has a pretty broad background in neuroscience and dysphagia and disorders such as ALS and Parkinson's disease and stroke. She is a speech pathologist by training. She has over 20 years of clinical practice experience, and she transitioned from patient care to research after seeing a need to improve patient outcomes. So that sounds really familiar to me. So I followed Dr. Lever's career for quite a while, following in her footsteps in a very small way. Her research is really translational. She focuses on understanding pathophysiology of dysphagia in neurodegenerative diseases across multiple species, so including humans and dogs, horses, and rodents. She's developed several forms of technology and methods to permit direct comparisons of dysphagia between humans and animals. And to date, she's had a continuous line of funding for her work, which has been provided through federal, foundation, intramural sources, including a current multi-PI R01 with Dr. Nicole Nichols, funded by NHLBI, and also an R21 through NINDS and also NSF funding. As a clinician scientist, Dr. Lever is really a role model for those who are really passionate about conducting translational research, and I'm so excited to hear her work today. Following Dr. Lever's talk, we're going to hear from Dr. Callum Ross, who is a professor of organismal biology and anatomy at the University of Chicago. He completed his PhD in biological anthropology and anatomy at Duke. His lab researches the motor control and biomechanics of the feeding system. So they use electromyography, fluoromycometry to study feeding muscle function, high-speed biplanar video fluoroscopy to study 3D tongue and jaw kinematics, and finite element modeling to study mandible mechanics. So to summarize, his research is really focused on understanding how both the jaw and tongue are moved and controlled during oral behavior such as drinking and chewing and swallowing. He has over 120 publications and a long history also of continuous funding from the NIH and NSF, including a current R01. I'm sure we'll hear a lot of his work from those funding opportunities today. So without further ado, I look forward to hearing your talks, and I'll be taking questions in the chat. And take it away, Dr. Lever.
Thank you so much for that terrific introduction. I really appreciate it. So I'm gonna, let me hit this again. Here we go. And now it's playing, correct? Okay, as I'm sure this audience knows quite well, Swallowing is a biological function that's necessary for survival across diverse species. It's therefore no surprise that dysphagia or swallowing impairment leads to a cascade of events that contribute to continued health decline. So to set the stage for this talk today, I'm going to provide just a brief overview about dysphagia from a human perspective, so to speak, and then turn our attention to laboratory rodents that are the models for dysphagia in my work, as well as companion animals. So dysphagia or impaired swallowing affects about 8% of the global population. It's primarily a disease affecting 60 years and older because of the growing population, the growing aging population in the United States. So some of the conditions affected are Alzheimer's, Parkinson's, ALS, stroke, head and neck cancer, and advanced aging. And because of this, dysphagia leads to life-threatening malnutrition, dehydration, and aspiration pneumonia, which, as we know, is a leading cause of death for a variety of conditions. Most people aren't dying because of the disease itself. It's because of the consequence of pneumonia. And this causes an ensuing cycle of repeated hospitalization over and over as people have recurrent pneumonia because of dysphagia. So dysphagia diagnosis in humans is a minimally invasive imaging method, one of two. We currently have two gold standards. One is fluoroscopy, which is called the modified barium swallow or video fluoroscopic swallow study, which I'll term VFSS throughout this talk. The other is endoscopy, which is the acronym is FEES, so fiber optic endoscopic evaluation of swallowing. So one is an X-ray modality, the other is an endoscopic imaging modality, but the bottom line is they're both minimally invasive, and we use these all the time in our human patients. But what is imperative to show you here is that in fluoroscopy, we typically see the entire oral cavity, pharyngeal space all the way down into the esophagus. But for endoscopy, we're seeing just a bird's eye view of this pharyngeal space. So it's a very limited exam, but we like in clinical practice to rely on both whenever possible. Regardless of the imaging approach, the goals are unanimous. They are to assess swallowing efficiency and safety, as well as to devise a treatment plan. So in normal swallowing for people, when we're looking for efficiency and safety of swallowing, you can see on the left here that it is a very fast swallow event, less than 1 second to transfer the bolus of whether it's liquid or pureed through the pharynx and into the esophagus. It's effortless, it's easy. There's no food or liquid residue. And importantly, there's no airway invasion, so no aspiration into the airway here. And if you look through the camera of the endoscope, you'll see something similar in that it's very quick, it's effortless, it's easy, there's no appreciable food or liquid residue, and there's no evidence of anything entering the airway, meaning aspiration. But what we will notice with endoscopy is you don't actually see the vocal folds at the time of the swallow because the swallow is obliterated by the epiglottis inverting as well as the light from the camera coming back. So dysphagia or in fluoroscopy, we're looking for impaired efficiency or safety in our patients. So on the left, it doesn't take much to recognize, or it's not difficult to recognize that this is a very slow, effortful swallow. There's lots of residue, and you'll even see airway invasion. And through the endoscope, you'll see something similar where it's very slow, it's delayed, there's effort, there's a lot of residue and airway invasion. But what I want to stress with you today is that dysphagia is also very common in companion animals. For most of the diseases and conditions that cause dysphagia in humans, we also see these same conditions in our animal patients. So we see in dogs, cats, horses, and just about any other species that would come to a veterinary center for help. So the major clinical problem across species is that dysphagia treatment is very symptom-based. We don't know the underlying cause, and without that known cause, we don't have specific treatments. So we end up doing what I would equate to a Band-Aid therapy, where we have altered diet consistencies, compensatory swallow behaviors. Again, these are just symptom-based. They're not getting at the underlying disease conditions. We place feeding tubes, tracheostomy tubes. Even in dogs, we do this, and breathing treatments for pneumonia and antibiotics also for pneumonia. So the problem is that there's such a huge knowledge gap. We don't understand how normal swallowing works. We know that there's numerous brain regions, and as Teresa Pitts says, there's this ever-expanding CPG for swallowing. There's at least 6 cranial nerves and spinal nerves involved, more than 30 pairs of muscles, multiple types of sensory receptors. So it's really a complex behavior. There are voluntary, reflexive, motor, and sensory components, and the sensory is the biggest culprit in my mind because we still at this point don't understand what the adequate stimulus is for swallowing. It's likely a combination of temperature, taste, touch, smell, and anything else that we can find as far as sensory in our world. So where and how does the neural control of swallowing break down for each disease condition is largely unknown. So the solution that I have been using is this One Health approach. So I'm trying to find commonality among species for the common good. So I have been, over the last decade, adapting the 2 gold standard dysphagia tests that we use in our human patients, so fluoroscopy and endoscopy, to be utilized with a variety of animal species, ranging from laboratory rodents and all the dysphagia models thereof, as well as companion animals. And so really the goal is that whatever we find from laboratory rodents can benefit companion animals as well as humans. And whatever we find on the human side can benefit companion animals and vice versa, so that it's not just laboratory rodents to help humans. So for today, I'm going to focus initially on laboratory rodent models and how using these 2 gold standard tests has helped me develop and identify rodent models that are appropriate for this goal. I'll also provide a sneak peek of my work with companion animals, and I'll always be referencing humans because I want to highlight my translational intent in this whole research agenda. So for laboratory rodents, fluoroscopy was miniaturized by a team of people in my lab that is now used on a daily basis with mice and rats. To do this, the goal was to promote self-feeding in a lateral view, but maintaining them in a restricted space so they wouldn't run away and do other behaviors. We wanted them to focus on just doing the behavior at hand, which we wanted them to eat and drink and swallow. So that entailed developing a lot of species-specific flavor contrast that we had to team up with pet food companies to do that. And in the end, we can do weekly to monthly testing without radiation toxicity. And here's an example of what we would see in our fluoroscopy test. On the left is a mouse, a healthy mouse that's drinking, and on the right, a healthy person drinking. And what you'll recognize right away is the sheer speed. The mice are about 10 times faster than humans in their swallowing ability, and rats are about 5 times as fast, or 5 times faster, I should say. And in slow motion, what you'll see is that the rodents really recapitulate aspects of swallowing similar to human infants, in that they would collect multiple attempts to collect a bolus of sufficiently large amount into this molecular space before a swallow is triggered. So we call this the swallow trigger point, this molecular space in the rodents. And here you can see in real time that the mice are very motivated to drink the contrast agent, and they willingly do so, and we can get a lot of data from them in a small amount of time. And this is all done at 30 frames per second, again, for translational intent to humans, where we are limited at 30 frames per second. So with this, we have also worked on objective quantification of video fluoroscopic videos in our rodent models. And so here's an example in slow motion where you can see every stroke of the jaw is tracked in the graph behind the scenes here. And the purple or pink line represents every swallow event. And every blue line is every pharyngeal transit time ending, so where the bolus has entered the entrance of the esophagus. So these, the jaw is tracked automatically by our software, and then we manually add in these other events. And then from that, we can get a whole host of quantified events of swallowing, ranging from lick rate, swallow rate, all kinds of inner lick and swallow intervals, lick swallow ratios, pharyngeal transit time. We can even follow the bolus all the way to the esophagus. And we're also moving into getting jaw open and closed distances and velocities to add more to our objective outcome measures. So what I like about this is that it provides an opportunity just graphically to compare two animals or one animal over time even. And so here's an example on the left of a healthy mouse and on the right with a mouse with ALS. So the top graph pertains to the healthy mouse, where you can see the jaw opening and closing as the licking for drinking is going on behind, and then the swallowing on top. And then you'll see in the bottom graph that it is a mouse with ALS. Right away, you can tell that the jaw rate is much slower. The amount of swallows, so the timing of swallows is very different, and the pharyngeal transit time is lengthened. So these are very visibly obvious differences when you see it graphically. And then there are numbers that the software provides to go with all of these measures. So with this objective quantification ability, we've been able to show that in this ALS mouse model, for instance, that at a young age, they are similar to their age-matched controls. So this is a mouse model of ALS that's a low copy number expressing. So it gets disease onset by about 6 months of age, which you can see in this graph where lick rate is already starting to slow. And then by disease end stage, which is somewhere between 8 to 10 months of age, it's very individualized in these mice, that lick rate declines quite a bit, and it's significantly declining as time goes on. We see this decline not only in lick rate, but also in swallow rate and other swallow metrics too. But what we can do in these in rodents, which we can't have the opportunity in our human patients, is at different stages of the disease, we can take tissues and we can take components of the brain that are controlling tongue motion and swallowing, and we can look at it for quantification. So here's an example just at end stage to show that the end stage lick rate and swallow rate deficits corresponds with tongue atrophy as well as hypoglossal motor neuron loss. And these are the lower motor neurons that are driving tongue movement. And these tongue-related deficits are early in this animal model, which really recapitulates ALS, where we find that tongue impairment is an early indicator in this disease. And so it's very similar to our human ALS patients in this way. And so this model is providing a unique opportunity for mechanistic and treatment studies that are currently underway in my lab. So to take this deeper, we also have been interested in swallow breathing coordination and discoordination thereof. So here's an example where instead of tracking the jaw, we can track the diaphragm relative to a fixed point on the vertebra. And we can see how the tracking behind here is the breathing in, breathing out. So inspiration, expiration, timed with each of the swallow events. And so with this, we're able to now start investigating objectively how swallow and breathing are coordinated or discoordinated. And you can see here that the majority of times this animal swallows, they're swallowing at the peak of inspiration or almost at the peak. And one time they're discoordinated. And so our question is this discoordination really increased in our animal models of dysphagia rather than healthy swallowing? But the caveat to this is these are at 30 frames per second. So to go even deeper, we've been doing chronic EMG recordings in our rodents. And this is an example of a rat who is implanted. This is a study that I do with Nicole Nichols. We implant EMG electrodes into the geniohyoid muscle as well as into the diaphragm. And then we can record in real time with video fluoro, where now we have a 20K sampling rate of what's happening at the level of the tongue EMG as well as the diaphragm EMG. And so with this, we can start to quantify how breathing and swallowing are truly coordinated or discoordinated, not just with swallowing, but licking also. And so we're excited about this new direction for us. So using this fluoroscopic approach that's been adapted from our human clinical patients to rodents, we've identified and established numerous mouse models and rat models of dysphagia. Some are spontaneous, such as biological aging. Several are genetic manipulations, like our ALS and Down syndrome, Prader-Willi, things like that. And then others are surgically induced, such as investigating facial nerve injury, laryngeal nerve injuries, things like that. And some are also reducible models that are radiation toxicity. We have several others that are also underway, but the common finding from all of these models so far is that they have impaired swallowing efficiency, but no aspiration. So the question is, why don't they aspirate? So for that, I've turned to endoscopy. And I see that there's a chat message. So I'm going to attempt to answer that right now. Oh, I think I'm going to wait for that.
I was just
Yeah, unless you can interrupt me, Alicia, if I should be answering something, I'd appreciate that. Thanks.
will do.
Okay, so for endoscopy, this is different from fluoroscopy because in fluoroscopy, these animals are awake, freely behaving, but for endoscopy, they have to be anesthetized, which changes everything. But I still think that there's value in doing this, which I'll be sharing with you. So for this test, these animals are lightly anesthetized, but deep enough that we're not abolishing the swallowing reflex. They are positioned in a custom fluoroscopy table that allows micromanipulator controlled use of the endoscope to advance it into the oral cavity. And you can see here how it goes into the oral cavity and in through the oral pharyngeal area all the way to the larynx. So this transoral approach really made us think a little differently about this, about rodents as a model and how this can really translate to our human patients and probably our clinical cases too with animals. So what's interesting is rodents are obligate nasal breathers, which means they are typically breathing through their nose rather than their mouth. When you look into the oral cavity with an advanced endoscope, you can see that you can see the tongue and the palate. As you get a little further, you can see where the epiglottis is here, but you don't see the vocal folds. So the larynx is hidden behind and underneath what looks like a very tightly coupled epiglottis and soft palate. And the only way to see this is if you depress on the soft palate, you'll see that the epiglottis actually comes out from this mucosal sheath that is holding it in place. So people don't have that, human babies don't have that. And so the question is, how does this change things from a rodent translational perspective to humans? The only time that rodents are breathing through their mouth is when they have some distress from a variety of health reasons. So when we see oral breathing, unless it's under anesthesia, we would assume that there's a problem. So here, when I'm using them in my research and anesthetizing them, they're not in distress because they're not awake and alert that they're breathing through their mouth instead of their nose. And their breathing resumes as they would have if it was through the nose, except for it is through the mouth. So there's no signs of distress when we do this. So how do we assess swallowing in these anesthetized rodents, you might ask? So I rely on mucosal application of citric acid. And when you look here through the endoscope, you'll see that when you're focused on this already coupled, tightly coupled epiglottis and soft palate here, where you don't see the larynx, you see a lot of tongue movement. So as there's inspiration, the tongue is moving out of the way, and during expiration, it's falling back to a rest position. So you can definitely tell when breathing is occurring versus swallowing under endoscopy. And we can record, this is just from a respiratory sensor on the abdomen, which you can see here, as well as a concentric needle that is positioned into the geniohyoid. And we can do this even in animals that are not terminal because it's just a simple needle insertion through the skin into the muscle that can be easily taken out for the animals to survive this procedure. So with this, we get respiratory sensor tracing as well as geniohyoid EMG tracing, and which shows you how stable they are. once we're decoupled and they're breathing through the mouth instead of the nose. So then what we do is we apply citric acid, which you'll see here, where I soak a little small micro bristle brush with 2.7% citric acid. I apply it all over the mucosa of the dorsal aspect of the epiglottis and in at the base of the tongue, so all through that vallecular boundary, and it evokes immediate, repetitive swallowing, hundreds and hundreds of times in a row. And you can see here that the tongue is actually punching down toward the soft palate rather than going the opposite way for breathing. And in this screenshot of EMG trace, you can see that the smaller EMG signals are breathing, and then the larger ones are swallowing. And with this, we can then start to investigate breathing, swallowing coordination, respiratory rate changes over time in response to being challenged with breathing, as well as any other swallow metrics that are based on rate and timing. And so we're in the process of developing algorithms to be able to distinguish healthy versus disordered swallow breathing coordination patterns in rodents using this technique. But we can also advance the endoscope further, just like we did in humans, where we can try to visualize the larynx for swallowing, but in humans it's obscured. So in rodents, we can keep advancing the endoscope through this decoupled epiglottic and velar space so that we can get a close-up view of the vocal folds. And we're probably 2 or 3 millimeters away from them for this view. And this is what we see. We see very easy breathing. I'm going to go back to here and show you again. So breathing where the vocal folds abduct apart, and then for swallowing, the vocal folds adduct, so they come together, and you can see that they meet at midline. And there's just this natural anterior glottal gap, which would be a ventral glottal gap in these quadrupeds. This is normal. And the way that instead of citric acid evoked, I should clarify here that just having the endoscope being advanced through that decoupled epiglottic and velar space is sufficient to evoke repetitive swallowing. It's not as fast and repetitive as citric acid, but it is still very frequent that we can get hundreds of swallows using this technique. So then what we do is we're interested in knowing, how do we alter airway protection? So for this, we developed a rat surgical model of recurrent laryngeal nerve, or RLN, transection and repair. So here you can see that we've transected the recurrent laryngeal nerve that's running alongside the trachea, or at around the 8th to 9th tracheal ring. And then it is sutured into this micro silastic tube to promote healing and growth across the gap. And it's about a 1 millimeter gap. And immediately after this happens, you can see that breathing is affected on the lesion side, as well as swallowing. And in slow motion, when you look at swallowing, you'll see that the airway does not close. The vocal folds have this very large glottal gap that goes from the ventral to the dorsal aspect of the larynx. So very unlike a healthy swallow. And then as we watch these animals over time, this is at the 4-month time point post-surgery, you can see that there's marked impaired secretion management. They're essentially looking like they're drowning in their own secretions. And this is showing evidence of aspiration, where you see the fluid actually running through the vocal folds themselves. And because of this, even though we don't see aspiration during video fluoroscopy with these same animals, we can clearly see that under anesthesia, they are very compromised in their ability to protect their airway. And now we're assessing the lungs for pneumonia as a biomarker in this model. We're also quantifying laryngeal function and laryngeal motion using some proprietary tracking software that we've developed here at the University of Missouri. And I've shared this before, where you can see that as the vocal folds are opening and closing, you can see the individual motion of of the right versus the left in the corresponding plot. And then when the airway closes, when the laryngeal airway closes because of swallowing, you see that they're going they're completely closing in the way that this makes it look like they're not closing, but when you look up here, they're actually oscillating around a central midline vector. And so therefore it comes out as that it's crossing that midline and going further, so that they are actually in synchrony in all of their motion. And then for here, this is an example of post-surgery. This is 1 month post as an example, where the vocal folds are not approximating at all during swallowing. And I'll play it one more time so that you can, appreciate it. There you go. So the vocal folds are actually both sides moving out of the field of view, and you see here that they're both going in the same direction. So this offers a way to start quantifying vocal fold motion relative to airway protection. And this is similar to how we do a test in humans where we're assessing the laryngeal adductor reflex. And in this case, we've got the endoscope into the top of the larynx, and we are delivering an air puff to one of the arytenoids. And when we do that, it causes an abrupt closure of the vocal folds. Other than this, there's no way to observe directly with endoscopy during awake swallowing in people. There's no way to observe the vocal folds actually approximating during swallowing. So this is the next best way to do it, which is evoking a different laryngeal reflex that is still related to swallowing. And when we do that, we can get a graphic display of the left versus the right sides, showing that it's a very symmetric response in humans, and we can get outcome measures that go with that. But the issue with rodents is that we cannot evoke the LAR reliably because the anesthesia actually abolishes or nearly abolishes this reflex under anesthesia. So it's not really a technique we can do with rodents. And that's why we've been proceeding with getting close-up viewing of the larynx during swallowing. So we've been investigating using fluoroscopy and endoscopy, investigating the effects of various treatments. We're doing neurostimulation, where we're injecting various opsins that'll go to the hypoglossal nucleus, and then we'll be selectively stimulating the hypoglossal nucleus to evoke repetitive exercise, so to speak, to retain swallow function and lick rate function in these animals. We also do things like exercise, and here's an example of a rat that's pushing at a known intensity against a force lichometer spout that is similar to doing tongue exercise in our human patients. We also look at a variety of effects of drugs like serotonin supplementation. We do gene therapy, and we look at other drugs like oxytocin effects on swallowing. So to briefly touch on a comparative to our veterinary cases before I close for the day. So we see a lot of dogs and horses from our local community that come to our veterinary health center at the University of Missouri. And what I want to point out is that dogs are nasal breathers primarily during sleep. So they're not obligate nasal breathers like rodents, and they're not necessarily quite like humans either, but they're kind of a combination of both per se. Whereas horses are obligate nasal breathers. If you block their nose, they cannot switch to oral breathing. So we have two very different species that don't quite match humans and don't quite match rodents either. So I want to highlight some differences with them. So using fluoroscopy, we've been trying to understand dysphagia in our companion animals that come to our clinic. And to do that, we had to adapt our human clinical protocol for video fluoroscopic swallow study, or VFSS, for dogs. In the past, they were tested in a lateral recumbent position and hand-fed or slightly force-fed, trying to get them to participate. And based on the poor outcomes using that approach, we developed a way that we can do hands-free testing in our companion animals in our clinic. And we can see self-feeding without struggling and assess actual natural eating and drinking behaviors. And here's an example of liquid swallowing and an example of eating where they barely chew, they swallow the kibble mainly whole. Using this technique, we've been identifying impaired swallowing and impaired swallowing efficiency and safety in our companion animals. Here's an example of a dog that came in from the community with a history of coughing and regurgitation, losing weight, and looking at thin liquid outcomes. You can see that there's a lot of residue in the pharynx after swallowing and aspiration. And for kibble, it's essentially a non-effective swallow where this animal can't even get kibble into the esophagus to swallow or to digest. So we're using the JawTrack software that we've been using with rodents to objectively quantify all of these videos in the hopes of identifying some common themes that would lead to a better understanding of dysphagia in dogs, as well as leading to our treatments. Similarly, we have adapted endoscopy for horses. We initially tried to use fluoroscopy, but it was really difficult because they're so huge and the field of view is so small. So we quickly moved to endoscopy. And the value of this in horses is that they don't need to be anesthetized or sedated in any way. So it's similar to our human patients, sorry about that, where we are just standing in front of the person. In this case, we're standing in front of a horse. We feed the endoscope into the nose and you can evoke the laryngeal adductor reflex just like humans. And we're excited about this in horses because they are very prone to recurrent laryngeal nerve degeneration. So here's a normal horse larynx, and here you can see laryngeal hemiplegia, which predominantly affects the left side in horses and primarily the larger horse breeds. Our goal is that in these clinical cases, we'd like to do earlier detection of laryngeal airway dysfunction. The way we do this is we don't do an air puff, and instead we found that horses have this really unique laryngeal wiring where we can just slap on the side of their withers, is what we call it, so that kind of like their shoulder, and it causes a either a unilateral or bilateral vocal fold closure. So it still is evoking the LAR, we can bypass needing to actually deliver air puff to the larynx and instead deliver the sensory stimulation on the outside of the horse. And we can track, just like in humans, the result of the motion. And here's an example where we have identified pathology where instead of adduction, the vocal fold on the affected side is abducting. And you can see that in slow motion where it's very desynchronous, and we can capture that graphically too. So what we're trying to do is with objective quantification using this software is to identify if this is something that could be really clinically useful in the equine clinic. So in summary, I wanted to kind of just bring it back to the beginning of, so what does species diversity really teach us? What we do know is dysphagia is common in people and animals. And because of this, it provides a really nice opportunity for cross-species research efforts to mutually benefit human and veterinary patients. But it's important to note that, we're comparing, maybe we could say they're both fruits. It's not apples to apples, but we're comparing two mammals in this case. We're comparing a rodent, which would be maybe a mouse with a dog, or a dog with a horse, or a human with a horse. So they're never going to be exact comparisons, but our goal is translatability. So with this One Health agenda, the goal would be to, across all of these animals, come up with a way to have earlier detection and quantification of impaired swallowing. Both efficiency and safety would be ideal, to identify underlying mechanisms that are driving dysphagia, develop targeted treatments, and then reliably measure the treatment response. And in conclusion, I just want to highlight that we know the tip of the iceberg about dysphagia at this point, and it takes a village to really accelerate research and translate findings to clinical practice in this really relatively new space in the scheme of things. So I'm really open to seeking collaborators throughout the CoBAD community particularly those that might have a One Health perspective, because I think together we could develop new and better rodent models of dysphagia that would be driving our identification of effective treatments or candidate treatments to try in our human and veterinary patients. And the ultimate goal is that we could have dysphagia move into precision medicine rather than symptomatic treatment. So I'd like to thank so many people in my lab, too many to name, my collaborators, especially Nicole Nichols, Ilker Osden, who helps with all of the neuromodulatory projects, and Felice Bunyak, who does all the computational analysis with me, and then our funding support that Alicia already mentioned. And so I thank you for your time, and we'll be happy to take any questions if we have a few minutes.
Great. Yeah, we definitely have a couple minutes before we transition. Just a couple questions. One from James Butler, who asks, plumbing of the nares to the trachea crosses plumbing of oral to esophagus, meaning you can't do both at the same time. I've heard this is not true in large cat cubs and in human preemies. How can you do both simultaneously?
Yeah, so my understanding from all the species that I've studied is that it's still one is occurring while the other is stopping. They're not going concurrently, because the way that the bolus flows, it still has to get past the airway. So like even in rodents where you have a tightly coupled velum and epiglottis, there's no way for a bolus that's in the pharynx to be entering the larynx. But when swallowing is triggered, it still has to go past the larynx. And so I think the advantage is that the larynx in several species and human babies is just much more rostral. So it's kind of hidden in a way, which provides some inherent protection. But I've never seen simultaneous swallowing without interruption of breathing.
Great, thanks. I just want to add, because I think it's an interesting topic, especially for this group, and thanks to Teresa Pitts for weighing in as well. But Jack Feldman had asked about the coordination pattern of swallows with the breathing cycle, and is there a phase preference for swallows? And Teresa had piped in and said, you know, in rodents, it's almost exclusively expiration. Cats and humans have a phase preference for expiration around 80%. I just wanted to know if you wanted to weigh in on that, or add anything you've seen in your studies that would contribute.
Yeah, we're seeing pretty much the same thing across the species, is that either right at peak inspiration or at the onset of the post-inspiratory phase that we do see swallowing occurring. It's unusual to see it at other times, but we're also seeing that we can perturbate that system in our models. So by causing some either genetic conditions or surgical conditions, we're trying to understand how that coordination is perturbated. And that's really the value of these models.
Yeah, and I'll just add that we really don't understand in a disease state how compensation will affect those phase patternings, and what's typical and what's optimal, and what we should actually be expecting, and what's disordered is still, there's so much to be teased out in that space. So just wanted to add that. Great. If there's more questions, feel free to add them in the chat. Hopefully we'll have a few more minutes at the end for more questions. But for now, we're going to switch over to Dr. Callum Ross, who's going to talk to us about animal models and swallowing in a different space. So go ahead.
Thank you very much. Can you hear me?
We can hear you loud and clear.
Great, cool. Thanks very much. So thank you for the invitation. Teresa, great talk. Really enjoyed it. As Alicia said, my lab works on comparative biomechanics of chewing and swallowing. And so Teresa's talk really nicely set us up for what we're going to talk about. So I'm going to be talking a lot about tongue and hyoid mechanics and function in vivo. I just want to start with a brief review of hyoid anatomy in humans. As we're all aware, the hyoid has got a greater horn and a body, and then a lesser horn on each side. And I've colored these for comparative purposes. So the greater horn is in other mammals is called the thyrohyoid. The lesser horn is called the ceratohyoid. The basiohyoid is the hyoid body. So if you just look at hyoid anatomy just in primates without going out into other mammals, there's a large amount of diversity in the shape of the basiohyoid, which you can see in yellow, and the degree of completeness of the chain connecting it to the base of the skull. And you can see these little colored bones here, and in the position of the basiohyoid at rest. So for example, here's a lemur hyoid up here, the second one down. The basiohyoid is a little bar. You have a complete chain with the ceratohyoid, and then the epihyoid, and then the stylohyoid on the base of the skull. Here's a galligo. When you get down to a macaque, which is like a human in some ways and not in others, you can see there's a large shield-shaped basiohyoid, an incomplete chain, as in humans, and the hyoid is positioned well forward at the back of the tooth row, unlike a human. Just at the bottom here is this weird extreme outlier, a howler monkey. If you go online and Google howler monkey calls, you can have a really good time listening to those. They have a massively inflated hyoid bulla that is positioned well forward like this. We recently published a review in dysphagia, and here we focused on the anatomy of the hyoid chain in different animal models. So for example, here's our macaque that we've been using in our lab. Here you can see the hyoid is positioned basically level with the back of the palate, which is similar to what it is in humans, except that in humans, the hyoid is positioned well below the mandible at rest, and in a macaque, it's up above the bottom of the mandible. And here you can see the diversity in these other animal models. I want you to just have a quick look at this cat up in here. Again, cats, like dogs, have got this complete hyoid chain. So there's a complete chain of little bones running from the base of the skull to the basiohyoid. And the question that, as evolutionary biologists, the question that we're interested in is, why does this diversity exist? Why are hyoids in different positions? Why is there different degrees of completeness in the chain? And how might that be reflected in diversity in swallowing mechanics? So I'm going to talk about two studies by graduate students in the lab. Dr. Courtney Osborne was a graduate student in the lab, and she studied monkey swallowing. And Pei Shu Lee is a graduate student in my lab currently. Has studied opossum swallowing, and we're just starting an exciting project with Teresa on dog swallowing. And Pei Shu's is available to answer questions in the chat. So if anybody has any questions, you can drop them in there. So let's talk about the first question: how do primates swallow? So this is the paper that we published a little while back, where we used XROM, which I'm going to talk about, and DICE-CT. DICE-CT is double enhanced contrast computed tomography. So you can stain soft tissues with iodine in such a way that you can do micro CT on them. So that enables you to get very high resolution images of soft tissue. So the basic question that we're going to address for the rest of this talk is, how is tongue-based retraction achieved? So first, what is tongue-based retraction? Here you can see a swallowing MRI. The hyoid is down in here. The tongue base is here. They're holding some pineapple juice up on the tongue, and then they swallow it. So let's go back here and have a look. The tongue-based retraction is when the back of the tongue extends backwards into the oropharynx, forcing the food bolus through the oropharynx and down into the hypopharynx. And so what you see here is there's the food bolus, and then the back of the tongue goes back to push it across. It's kind of subtle in humans. But the point I want you to kind of note right here is that the hyoid bone, which is here, as that tongue base retracts, the hyoid goes up and forwards. There's the hyoid there. How is that tongue base retraction produced? What is the mechanical mechanism that pushes the back of the tongue back? There's a couple of ideas. One is what we call the extrinsic hypothesis, that posits that extrinsic tongue muscles with posteriorly directed vectors of action pull the back of the tongue back. The other idea is an intrinsic hypothesis. It's the muscular hydrostat model. And a muscular hydrostat proposes that the tongue is essentially a closed liquid volume. And so if you narrow its dimensions mediolaterally, and then as a result, you will get deepening and the tongue will get taller, or it'll get longer, or both. So by changing the shape of the tongue using intrinsic muscles, for example, you could get tongue-based retraction that way. So let's just have a quick look at the human anatomy. Here's two of the muscles that have been purported to be involved in tongue-based retraction in humans. Here's the styloglossus muscle. That's got a posteriorly directed vector, and as does the hyoglossus. Palatoglossus is another muscle that's been accused of being involved in tongue-based retraction. And in this sequence of figures from Gray's Anatomy for students, you can see they posit that palatoglossus and hyoglossus are important in pulling the back of the tongue back. But is this true? We're really interested in what's happening in the middle of the tongue. And I do want to point out that palatoglossus, hyoglossus, and styloglossus all attach to the side of the tongue. So I think we should be interested in the question of whether if you pull back on the side of the tongue, why was the middle of the tongue going to go back even further? You can tell I'm suspicious. So we've been working with macaque models. This was some video from a 2D study we published a little while back. And macaques are, we've got some tantalum beads implanted in the tongue. And we have them, now we have them implanted in the mandible and cranium as well, instead of these screws that you see here. Here's a grape coated with barium, quite delicious. There's a swallow. And you see multiple swallows here. Here, there's another one. You can see the bolus accumulating in the valleculae. See the soft palate contacting the epiglottis, about there. And so these macaques achieve tongue-based retraction also. This total obscuring of the oropharynx back here is because the back of the tongue is pushed back and the back of the pharynx comes forward. How is tongue-based retraction produced? So we've been using this workflow called XROM, X-ray reconstruction of moving morphology. And in XROM, you take two video fluoroscopic or video radiographic movies at the same time in a calibrated space. So here's one X-ray source, here's one image intensifier. If you can convince the animal to do the feeding in the space here, then you can get these two movies from different perspectives. And in a calibrated space, you can then reconstruct the movements. So in order to do this, we implant small tantalum beads. 1 millimeter is usually what we use. You can get 1 millimeter beads that have been laser drilled down the middle, so you can suture them into soft tissues. There are 5 millimeter beads, but I can barely see my phone when it's at the end of my arm, so the prospect of me working with 0.5 millimeter beads is remote. But you can see these tantalum beads embedded in the tongue. Tongue tip, we've got some mid tongue and we've got some posterior tongue. And here's the view from the side, and here's a view from above. So this is a study. We're also recording from the brain. That's why you see these rather kind of these wires coming out of the top of the head. We're recording from the brain as well, and we just published a paper on that, how brain activity is related to tongue shape. But so here we are taking two X-ray movies of this monkey at the same time, tracking the beads in 3 dimensions. Then with a CT scan of the animal afterwards, you can then reconstruct the movement of the mandible, the movement of the hyoid, which is the shield-shaped thing right here, and then the movements of the beads embedded in the tongue. And there you can see those beads moving around in 3 dimensions. So let's look at some data. There's a bunch of data on the left-hand side, and we've got some diagrams on the right. So here is the gait time profile. Here the jaw is closed, and then here at minimum gait, that's the start of slow open. Here we see the jaw slowly opens, and this is where the tongue-based retraction occurs. The mandible is closed, but it slowly opens during the swallow. Here is the period of tongue-based retraction in this light tan, almost impossible to see rectangle running all the way down the plot here. That's the onset of tongue-based retraction, and that's the offset. So when this palatoglossus, this posterior tongue marker in the midline goes backwards, that's the start of it. And when the vallecular marker stops going back, that's the offset. We've got a few traces here. The oral cavity volume, which we estimate. Here, we show that the oral cavity volume decreases during the start of tongue-based retraction, and then it starts to increase as the jaw opens. We've got the posterior oral tongue volume, which we measure, and what you see is that the posterior oral tongue increases in volume, as does the tongue base at the beginning of tongue-based retraction. All right, so let's have a look at this intrinsic hypothesis. If the intrinsic hypothesis is correct, then we would expect lengthening of the tongue, posterior lengthening for the tongue base to go back. We would expect that to happen, and it doesn't really very much. It kind of gets longer. And we would expect that to be associated with narrowing of the tongue. That's the green areas. We would expect, in contrast to what you see here, we would expect the tongue to be narrower and perhaps getting more shallow, and that would cause the back of the tongue to jet out into the oropharynx. That is not what you see. Here is the depth is the blue trace, the length of the tongue is the red trace, and the green trace hiding in the back is the width. During tongue-based retraction, when the tongue mark, this posterior tongue mark is going back, the length of the tongue is almost constant. The tongue is actually getting deeper rather than shallower, and the tongue is getting wider at the beginning of the tongue-based retraction rather than narrower. But that doesn't look like that's a very good model. Down the bottom here, you can see the hyoid movement during tongue-based retraction. It's going forward, that's the X, and it's going upwards slightly during tongue-based retraction. Let's talk about these muscles, this extrinsic hypothesis. For the extrinsic hypothesis to be correct, during tongue-based retraction, this period here, you would want to see palatoglossus, styloglossus, and hyoglossus shortening. The green trace is styloglossus. It shortens slightly at the beginning of tongue-based retraction and then not at all. Palatoglossus does not shorten during tongue-based retraction. Hyoglossus does, but that's a red herring. So let's talk about the kinematics of these markers during tongue-based retraction. So you can see here's a mandible with a tongue, and you're looking from above. Here's the hyoid, and here's these different colored markers that match the box plots that you see here. 0 is these markers not moving forward and back, and when they protract, they move to the right, and when they retract, they move to the left. These gray boxes in the bottom left are the midline tongue markers. This is the tongue-based retraction. This one here goes back furthest. That's the superficial posterior surface marker. This is posterior deep, and that's the follicular marker. So these gray boxes are telling you, gray and white boxes, these are telling you this is tongue-based retraction. This blue box here is this marker on the right side of the posterior tongue, and the purple one is this marker on the left side of the posterior tongue. And what you will see is they are not going backwards. Tongue-based retraction in macaques is a central tongue phenomenon. The sides of the tongue don't go back. And so one has to ask oneself, if the sides of the tongue are not going back during tongue-based retraction, how can it be that the muscles that attach to the sides are the ones that are pulling the back of the tongue back? Just to put in a little more, just to focus in on it, here we see this purple box represents the movement of that marker on the side of the back of the tongue, and this blue one is the side of the back of the tongue here. And you see the middle markers are going backwards, but the markers on the side are not. Here's the tongue kinematics. We measured the length of these muscles during tongue-based retraction. The purple is the palatoglossus. It's right at 0. The palatoglossus does not shorten during tongue-based retraction. The styloglossus does not shorten during tongue-based retraction. The hyoglossus does shorten. But the interesting thing about hyoglossus, it's not pulling the tongue back, it's pulling the hyoid up. And here you can see during tongue-based retraction, this is the hyoid displacement. There's a lot of protraction and there's a lot of elevation. I talked a little bit about how the back of the tongue increased in volume, and that is what you see here. We kind of calculated these 3D volumes of the tongue base and this posterior tongue volume here. And what you can see, if you just compare these two cartoons based on real data, here is the shape of the tongue at the start of tongue-based retraction, and here is the back of that part of the tongue after tongue-based retraction. All of this volume that's in the tongue is kind of being squeezed back into the tongue base. That is why the tongue is getting longer, but it's not due to the muscular hydrostat model, because the muscular hydrostat model, let's just go back up here. Muscular hydrostat model would predict that the tongue would get shallower and narrower and then longer, but that is not what you see. The tongue actually gets taller, if anything. The width gets wider, if anything. So the muscular hydrostat model doesn't apply. Those lateral muscles aren't pulling the tongue back. Instead, what we propose is this hydraulic model. Look at that hyoid going up and forward into this, what we're calling the oral volume right here. And there, that tongue-based marker gets pushed back into the oropharynx. We believe that what you're seeing here is, here's our hyoglossus, geniohyoid and mylohyoid, and they're pulling the hyoid up into this almost closed box, squeezing the middle of the tongue back into the oropharynx and pushing the food with it. So we propose this hydraulic model of tongue-based retraction in a monkey that we also suspect applies in a human. So you take that hyoid and you jam it up into this closed volume, and then the food and the back of the tongue get pushed out. So that's how macaques swallow. Macaques do not use a muscular hydrostat model, and macaques do not pull the back of the tongue backwards by shortening those muscles that attach to the side of the tongue. Instead, they use this hydraulic model. So that's true of macaques. We suspect it's true of humans. Is it true of other mammals? I talked at the beginning about how there's all of this diversity in hyoid morphology, hyoid chain completeness, basihyoid shape, and the position of the hyoid. And how is that related to diversity in swallowing mechanics? So this is a lateral view of an opossum. Here's the basihyal here. And then here's our thyrohyals, and there's the larynx. You also see, those of you, obviously most people here interested in breathing, there's also variation in the position of the larynx relative to the hyoid. And that is also related to soft tissue anatomy here. And you saw some of that in Teresa's talk. There's a lot of variation back here in the oropharynx and laryngopharynx, related maybe to hyoid anatomy. What does that mean for the mechanics of swallowing and breathing? And what does it mean for the use of animal models in swallowing and breathing research? So we put opossums in the X-ROM. So here's the two simultaneous movies of an opossum chewing in a box. The great thing about X-ROM is you can put the opossums in a dark box and then just induce them to feed. Here is a reconstruction of this with the coordinates of the markers in the cranium and the mandible and the tongue. You can animate the cranium and the mandible and the hyoid as these animals feed. Here's an opossum holding something in its hands and pulling it off with its jaws and swallowing. So I'm going to present some data on opossum swallowing that Pei-Shu Lee has been collecting for his dissertation. See if I can get this animation to work. Okay. So this is a view of an opossum swallowing from above. The mandible's going up and down. The hyoid doesn't seem to be moving very much. That's what you see in yellow. Look at that red marker. The red marker is the tongue base. So there is tongue base retraction in opossums. That's what that poke when it's squirting back like that. But look how the hyoid is barely moving. It's not like a macaque where the hyoid gets jammed up into that oral volume and the tongue base goes back. That's not what you're seeing here. Instead, it looks like there is some maybe lingual deformation is consistent with the muscular hydrostat mechanism is what we're seeing. So this is a dice CT view of an opossum. It's a little strange view. You kind of, it's posterior and lateral on the right side. So you're kind of looking out through the animal's mouth. Here, the tongue is sticking out. And what these arrows are showing is the idea that if we narrow the tongue, these blue arrows, if we squeeze it sideways, maybe we can we could make the posterior tongue taller, but if we didn't do that, we could squeeze the back of the tongue back. That's intrinsic tongue muscles producing this tongue-based retraction. And so I'm going to stop this because it's distracting. And so here's the actual data. So during tongue-based retraction, when that little red marker is going back, you see that the width of the tongue, the back of the tongue decreases. And you see the tongue base height is increasing slightly, but it's basically constant. And the tongue base length increases. This is the tongue base retraction here. As the tongue lengthens, the tongue base goes back. And so these data in the opossum are consistent with a muscular hydrostat mechanism. That is to say, to make the back of the tongue go backwards into the oropharynx, we squeeze the tongue sideways. We don't allow it to get taller. Maybe that's what the hyoid's doing, is holding it in place. And then the back of the tongue squirts out and the food goes with it. I'll move on to the next slide. Okay, so the interesting thing here, so tongue-based retract, what's the mechanism? So we think that this muscular hydrostat model has got some legs. Just to give some idea about the differences between the macaque and Didelphis. Remember, in the macaque, the oral cavity volume was this space up in here above the mylohyoid. It actually decreases at the start of tongue-based retraction in a macaque. So you have a decreasing tongue oral volume, the hyoid's being jammed up into it. And that's kind of what's generating this force to squeeze the back of the tongue back. You don't see that in an opossum. You see the oral volume here basically static. I mean, this shows it's increasing. We've been working on different ways of estimating the oral volume, and that there's definitely some tricks there, but it's essentially static in an opossum. So if the volume of the kind of oral volume, as we call it, is constant in a didelphis, that also supports the idea that this is not a hydraulic mechanism of tongue-based retraction in an opossum, and it supports the idea that it's a muscular hydrostat. We'll just look at a little bit of data here just to kind of present this in another way. Here's the gait. So this is the gait time plot at the top. During slow open, as the mandible opens slowly, it's pretty fast in this opossum, but this is the period of slow open when tongue-based retraction occurs, and this is tongue-based retraction period here. The hyoid is basically static. I mean, this is the start of tongue-based retraction here. Yes, it's going up a little bit, and maybe it's going AP a little bit and up and down a little bit, but it's very small displacements compared with a macaque. Here's the palatoglossal midline posterior marker. This is the tongue-based retraction kinematics itself here. And this is the oral volume right at the bottom. Look how static it is. So the hyoid isn't doing much, isn't driving this tongue-based retraction, and the oral volume is basically static. Let's look at these tongue markers. So again, let's just reiterate what we saw in the monkey. In the macaque, these gray and white and dark colored boxes here, these are these midline tongue markers that get squirted back into the oropharynx. But the markers on the side aren't moving. There's some shear planes in the tongue. And here's didelphis. The didelphis hyoid is further back. This is Euroline is the back of the palate. So the hyoid's further back. During tongue-based retraction, you see a very similar phenomenon in the didelphis. That is to say, you don't get much retraction of these lateral tongue markers compared to the midline. So looking at these data, and we need to collect more data from more opossums. So these are in a sense preliminary, but we've got a fair number of swallows here and the pattern is the same. So there is tongue-based retraction in opossums. So opossums might be a good model for studying mechanisms of tongue-based retraction. They don't seem to use the same mechanism as macaques. There are similarities. The sides of the tongue are not moving posteriorly very much, but the middle of the tongue is. So there's some similarities between these species in the sense that during tongue-based retraction, the middle of the tongue kind of moves relative to the sides. So we really need to understand the mechanics of how that movement is being produced within the tongue. So looking at different animal models would be a good way to study that. If you look at didelphis and macaque hyoid bones, they have different shaped basiohyals. Maybe this large shield-shaped basiohyal in the macaque is important for having a large surface area to drive this hydraulic mechanism. Maybe in didelphis, the absence of that shield shape is related to the fact they don't use a hydraulic mechanism. They don't have a complete chain going up to the skull. So, and the position of the basiohyal on the macaque is well forward. The position of the basiohyal on the opossum is all the way back here, like a dog or a cat. The position of the basiohyal isn't related to the degree of completeness of the chain. And the degree of completeness in the chain doesn't seem to be related to the mechanism of TBR. So there's complexity here that we don't understand. We don't understand why there's this, we might understand why there's variation in basiohyal shape. We don't understand why some animals have got a complete chain and others don't. We don't understand the mechanisms that animals use for tongue-based retraction and swallowing, except in these two animals, and they're different. So the diversity that you see in morphology across animals, opossums and dogs and macaques that we're looking at here, this diversity in morphology of the hyoid in some sense might be related to differences in mechanisms of swallowing, but it might not. And so the take-home message here is that there is a lot of really interesting, fascinating diversity out there that we, in non-human mammals, that we can take advantage of to learn about the basic mechanics of swallowing and breathing. And I put this dog up here just to remind me to say that Pei Shu and Teresa and I are starting a collaboration on XROM of swallowing in dogs, and that's going to be exciting. I'm looking forward to that very much. So thank you for your attention.
Great, thank you so much for that, Dr. Ross. We have a couple of questions. One from Jack Feldman, who asks, could dysfunctions in swallow play a role in obstructive sleep apneas? And do patients with dysphagia have an increased incidence of obstructive sleep apnea?
I'm going to bow out of that question because I don't know the answer. Thanks for asking.
Yeah,
I
I
don't-
actually put a brief response in the chat that
Oh, yep.
it's hard to know which is the chicken or the egg in this case,
gonna say
but
that.
we do have literature saying that patients with OSA are also prone to signs and symptoms of dysphagia. So we don't know if it's exactly what's the underlying cause, but there is that link there. So both impaired swallowing efficiency and safety. So aspiration is an issue, as well as just the whole biomechanics of swallowing can be affected.
Seems like the literature is very correlative at this point, where just showing kind of the incidence rates, but the mechanism underlying is really unclear at this point, and I mean, subject to investigation for sure.
Since-
Bill
can
Milsom.
I interject just quickly? Teresa, a lot of sleep apnea patients with certain phenotypes have very enlarged lungs, or tongues, excuse me, lots of fat content in there. And so that's part of the obstruction that occurs there. That should impair swallowing alone, should it not? Unless,
Well, if it's obstructive in the sense of in the act of swallowing too, then yes. If it's not preventing, so you can still have, say, a very enlarged tongue, but if you can still get the bolus to be swallowed the right way, that is not a problem. So I wouldn't say that just because it's a large tongue that would be dysphagia.
Yeah, where is the fat accumulated, Gordon? Is it distributed throughout the tongue or in specific places? That would play a part too, don't you think, Teresa?
I'm by no means an expert. We may have some on here. I think I saw some earlier, but it's more distributed through it. I mean, it actually deposits a lot there. And you know, what's chicken? You have the same chicken and egg problem as the sleep apnea because of the fat tongue, or does the sleep apnea cause the fat tongue? And the answer is probably yes.
Yeah, it's probably both. Chicken
So-
and the egg.
Just to talk about, just to kind of talk a little bit about the tissues that make up the tongue. One thing that we are interested in the lab is not only the muscles and the connective tissues between them, but that lingual tonsil that's on the back of the tongue. It's pretty stiff. I mean, when you dissect human cadavers, it's, you know, it's not moving very much. And we wondered whether it might be like a piston, right? The tongue base moves back, and you have this lingual tonsil gets pushed back. So the material properties and textures of the tongue definitely are in need of study.
I think it's a good time to add, if anybody else has questions or wants to weigh in, please feel free to unmute and bring your camera up and just jump in with discussion. We have another question from Bill Milsom. He says, looking at Teresa's x-rays, what jumps out is the curvature of the spine. I suspect that the only time the spine is not curved to this extent is when animals are curled in a fetal position. Does this imply that the trachea and esophagus would also be less curved and that breathing and swallowing easier?
That's an interesting question. And so in x-ray, we test the animals in a couple positions. So I could comment on that. I've not looked at them under x-ray if they're curled in a fetal position and doing other crazy things, but I see them while they're grooming, while they're postured for chewing and swallowing. So they're more upright and hunched over. And certainly it seems as if it's the spine, surprisingly, in the ALS models, they end up very extremely curved throughout most of their adult lifespan. So there's not a whole lot of motion with it, but with the healthy animals, we'll see more motion, obviously. So to answer your question, I don't think I really can, except to say that the cervical spine doesn't seem to be as I guess, altered during all of these behaviors as the rest of the spine. And so for the oral pharyngeal phase of swallowing, I don't think it would be, that's, you know, not of anything of concern, but of course the esophageal and the GI tract thereafter, as well as the effects on the pulmonary system may come to play when you start talking about the spinal position.
Yeah, part of the reason I ask is years ago doing side x-rays just to see where electrode placements were with brain electrodes and EMG, EKG leads. Noticed in our animals the exact same thing. So you lie them flat on a table and you expect their spine to be flat, and it's not. It's got this massive curve in it. And so the only way you actually get that curve out is to put them in a fetal position. But it also explains in part why when the animals are anesthetized and limp, that's the position they go into.
Mm.
And if you think about it, to be in that position lengthens the neck.
Mhm.
So to be able to do that, you have to have that long neck, which means that when you're in the upright tetrapod position, you've got that natural curve. It's just
Mhm.
intriguing.
It is.
We don't think about it as bipeds.
No.
Bill, one other thing that we know, at least from humans, is that position changes swallow. So that could be neck afferents, vestibular afferents. And so there's some studies where they put them in tilt tests or move their head up and back. And so, it could be coming spinal feedback, but we know the position of that neck and the feedback that comes from the position, the body position in space and where the neck is, does have a pretty significant impact on feeding.
That's a great question, Bill. Peishu has a paper in I think it's accepted, at least in revision, where he studied the effect of in the opossums, the effect of head-neck angle. So this is, my hand is a head and this is the neck. So the effect of flexion of the head on the neck and gape at the same time. So here I've actually got an opossum. So we have 3 different gapes and we have different head and neck positions. And those 2 factors impact hyoid position. The head neck angle affects it the most. Bill Peishu will correct me if I'm wrong. And as Teresa Pitts said, that's also true in humans, hence the chin tuck maneuver, right? And so again, these animal models, I think, might give us a way to study head neck angle and its effect on swallowing. Peishu, you want to leap in and correct me?
No, I was just going to add that when we actually measure the kinematics of the hyoid during swallowing, for possums swallowing at different head postures, going from sort of the more neutral to a more flexed posture, there's actually a lot of stereotypy in how the hyoid moves, both in terms of excursion magnitude and in terms of the timing of, say, maximum protraction, maximum retraction, etc. So it does definitely seems like, well, either one of our hypotheses is that there is some kind of sensory efforts that the animals are taking in and adjusting in real time how the position of the hyoid is controlled, whether that's vestibular, whether that's some other kind of sensory afferents, we don't know at this point, but that's one of the hypotheses that we think could be happening.
If I can just jump in and say something about babies, because that's all I know about. Infant animals often have a preferred position, which we call downward facing pig and upward facing pig, and which to me suggests that it is more about afference than it is about the biomechanics of position. Because if it was position, every baby animal would want to be the same way. But we've got animals that have their head down and animals that have their head up. And we got animals that lie down and have their necks so extended to eat. And they're just a large number of different things. And I want to say, as long as I've got Peishu here, I am going back to my baby possum. You've inspired me. I'm going back to I have baby possum feeding data from 25 years ago. And I'm actually going to go back and look at it and do some of the same measurements that you've done on that to see what we can see.
I'm super excited to see what we find out, Rebecca.
I know Dina Hallen was doing something similar to this in locomotion, right? Because they were looking at cats that were walking downhill versus uphill with a collaborator at Georgia Tech. And they destroyed the vestibular nuclei and saw no change in the corrective positioning. So I'm betting the vestibular might be part of it, but it's certainly not the whole story for a lot of this. I think those neck afferents do a lot. And so I would bet there's probably pretty similar mechanisms for locomotion as there is for feeding.
Any other questions from those in the chat, or anyone want to pipe in with any thoughts from the audience? Otherwise, we can wrap up a little early. I just want to thank Dr. Lever and Dr. Ross for keeping their presentations concise and allowing for some extended time for discussions and chatting about these topics. It's always just a great pleasure to hear from you both, and thank you so much.
And thanks to you all for tuning in. Thank you, Teresa. Thank you, Callum. Thank you, Peishu. And thank you, Alicia. Great job. Enjoyed. All
Happy
right.
Thank you.
Friday.
See you
Thanks,
next
Have a
time.
good weekend.
Bye.
Bye-bye.